from here at the University of Adelaide, who is looking at investigating the molecular basis of echidna biology for the application in conservation, wildlife biology and habitat monitoring. Please welcome Frank to the stage.
I'll wait. That'd be really good. Yeah, thank you. Okay, thank you. Okay, well, thanks to the organisers, and I'm a bit more excited than I usually am, and I've flicked the slide already, you can see. So let's see, go back, because I want to talk about something that we've sort of been working quietly on for a number of years, but we've recently received some funding from the Environment Institute that really enabled us to scale this up. So really, for many years we've been interested in, this is the pointer, right? Is the pointer here? No, yeah, okay. Got this worked out now. Change the slide again. So really what we've been, what our long-term goal is to investigate the molecular basis of monotreme biology, and they got a really fascinating biology, and I can talk to you about this for hours. But I think what we're really keen as well is to use some of the knowledge that we've gained, genetic and otherwise molecular, to actually use this, also give back if you like, and use it in the context of conservation, captive breeding, captive management, but also in ecology research in the field. So I'll give you a bit of an example and overview of where we're at in terms of molecular work on these fascinating species, and then give you just a few snapshots where we've started to get into, into taking some of that research into the field, if you like. So, monotremes, without doubt, are my favorite Australian mammal. I think they're fantastic. For evolutionary biologists, they basically represent the earliest living offshoot in mammals. And we've been using that fact to gain some fantastic insights about the evolution of the mammalian genome, but mammalian biology in general. So you can see the mammalian group by the monotremes, or the egg-laying mammals, which contain the platypus and the echidna. You've got the marsupials and the placental mammals, or eutherian mammals. So it's been particularly useful in comparison with birds, for example. So it's the evolutionary position that makes them really interesting, and there's loads of fascinating biology in monotremes that we still need to understand. And last but not least, they're iconic species. The echidna is found all over Australia, also in Papua New Guinea. The platypus only found in Australia. So we have these animals here in Australia, and I think that really it's important that we also lead an effort to understand them and also protect them. In South Australia, I think in particular, monotreme research has been very strong. In fact, it was Willem Haag who reported on the same day from the South Australian Museum as William Colville was running through the bush chasing platypus in Queensland, and they reported at the same day in 1884, almost 80 years after the first samples were sent to England, that in fact they are egg layers, they lay eggs. So Haag, they discovered that an egg in the pouch of the echidna right here in Adelaide. So I think that's a lot of important history. When I first started working on these species, my background is sex chromosome biology. I started realizing that for the molecular biology, and if you basically fast forward now, to nowadays, has been limited by access to material. Whenever we got an opportunity to get some material, we're really trying to do most out of that. And doing that for well over 10 years, we've really assembled a worldwide unique collection of material that's to be used by researchers. And we've had some fantastic collaborations through that, and some very nice papers. And I think we've been able to basically establish international reputation and leadership to connect the molecular biology with the biology of these fascinating species, and that includes the Platypus Genome Project. It was completed in 2008, but it's still an ongoing effort. So that reminds me that with particularly attention today on the echidna, the other monotreme, and while we can gain a lot of information also about the echidna having that genomic base in the platypus, we're still catching up a little bit with the echidna, but the information is in the process of becoming available. So we'll be in a position to do similar things in the echidna. And as I said, the ongoing interest of researchers really leads to constant improvement of the molecular resources that are available. And just to illustrate, how this has changed the way we can do research. When I started in monotremes and we wanted to isolate a single gene, that could well be meaning 1 year work in the lab or 1 year of a PhD student in the lab. So when the Platypus genome was published, and I looked in the public database the number of genes that had been published or deposited in the public database 30 years before the genome project started, was 23. And after 4 years of genome project involving about 100 researchers, so a huge effort, it's about 18,500 gene models, and that number is probably not quite accurate. Pretty much the number that we expect is the gene number in mammals. So basically a fantastic resource, and rather than working for a year in the lab, you could go onto the computer. You had a pretty good chance to find your sequence of interest. So really the interest in MarLab is, you understand, as I mentioned before, the extraordinary biology. We've also been using this in evolutionary sense to compare that with human disease. We've got projects, and I won't go into any detail, but as I mentioned at the beginning, I think now we're really trying to translate this knowledge into conservation biology, captive populations, and use that in the conservation context, developing tools and then taking it into the field and actually apply this. So I think the history, the material that we have, the connections that we made, but I think also some unique opportunities here in South Australia. I really try to justify that we want to have a leading effort here in South Australia to address these aims, and again, for the purpose of this talk, really build a solid platform in which molecular biologists can communicate with field researchers and zoos to use the new molecular information in the context of conservation and ecology. I think another a reason is that you may or may not know that the Kangaroo Island echidna population has recently been declared endangered. Would be the only echidna population in Australia that's officially been recognized endangered. Echidnas have in many places declined quite rapidly, and that's the bad news. The good news is that with Kangaroo Island we probably got also the best study echidna population, which is due to long-term fantastic work that Peggy Rees-Miller has done in Kangaroo Island. We work very closely together with Peggy. So there's clearly the need to have conservation, and we also have the information about this population available. So I think there's really a lot right here in South Australia that can be combined. So I want to now go on to snapshot number 1. And that's we had a great conference here in 2008 after the publication of the Platypus genome. And the aim of the conference was very much to bring together researchers from different areas and communicate with the gene jockeys, if you like, and see where the collaborations could be. And so I've talked to Arthur Ferguson from Perth Zoo, and Perth Zoo has been very successful in actually captive breeding echidnas, and here you can see these wonderful puggles. And he said, if you look at an echidna, it's not actually very easy to, or you can't tell whether it's a male or a female, or very few people can. But we would really like to know, because it would make a difference in terms of how we manage them and how we keep them. So he said, well, we've been working on their sex chromosomes for a long time, so surely we can find some sequence to determine whether these little guys have a Y chromosome or not, which defines males in mammals. So he said, you plug out some hair, that's not very invasive. So we got some hairs, we extracted DNA from the hair samples, and then used some of our Y chromosome sequence to determine whether they're male and female. And we've done that routinely now over the years. And I think it's a fairly simple example where, for us it's not a lot of work doing the extraction and the PCR, although it's also not it's not trivial, it's very little DNA, but actually having an impact into how the animal is managed in the captive breeding context, and we're keen to continue that. Another good example of a collaboration that has been sort of ongoing, although it's been sort of, we need to, I think, revive this collaboration. It's been a fantastic opportunity that we have here to collaborate with Zoos SA, which includes the Monarto Zoo, at the zoo here in Adelaide. Adelaide, there's a number of echidnas on display, and then at Monarto Zoo, and that's a map here, basically there's a fence around the wild echidna population, and there's a very enthusiastic group of volunteers that sort of keeping an eye on the echidnas. Again, Peggy Rees-Miller was very enthusiastic to compare the biology of these Monarto echidnas, for example, with the work that she's done on the echidnas in Kangaroo Island. So we got a well-studied, and if well-studied I mean in terms of vegetation, in terms of insects. It's really interesting in Monarto because it's a mixture of native vegetation and revegetated areas, and a lot of work has been done on invertebrates in these areas. There's excellent facilities, and we've had a lot of support from veterinary personnel, from the volunteers, and we've basically had a pipeline where if an echidna is sighted, we could take some samples and we could process this in the lab on the same day. So I think it's a really good opportunity, and now we've actually got some funding to really scale this up. So I think that's again another local good arrangement that enables us to connect these two areas, molecular biology and the wildlife biology. Another area that I find that we haven't started doing work on, which I find very exciting, is that we have now technologies to really probably use the echidna as an environmental indicator and cultivator. So from Peggy's work, we know that a lactating female could be foraging for up to 18 hours, and she's counted up to 1,000 digs. So contrary to their reputation as sort of living in the slow lane, you can see that echidnas are actually very active animals, and that's of course because of the high demand through lactation that they need. So they would consume a considerable amount of invertebrates, termites, grubs, larvae, ants, beetles, and other things, and identifying those species using some of the new sequencing technology. And Emma was, I think, giving very nice examples in the marine context how this is now used, and can actually give us a very good indication of what's in the ground. And from Peggy's observations also shows that echidnas are actually able to utilize areas where there's more of these invertebrates, so a very nice reflection of the actual abundance, but also of course being useful in controlling these invertebrate populations. So what we like to do is to collect echidna droppings and really establish, using some of these high-powered sequencing technologies, to get a very comprehensive insight of the diet, but also this will reflect then what's in the ground, and that of course can be very important for landowners, and in natural parks that can be very important to obtain that knowledge. To summarize, I think we got a really strong connection here, having those resources, the knowledge, the links, and that's very much an international effort. We collaborate with many world leaders in molecular aspects of this worldwide. We got links to zoos, and we got these populations in South Australia, but of course that would also include other populations. And in the past we found that this constellation of connecting the lab with the wild is fantastic in terms of community engagement. We had high school students going up doing work, and the idea is to really have exchange in samples and information between these two areas, and really gain more information about their biology, but also see how they fit into the environment and what information they can reveal about the environment. So it just leaves me to thank people in the Lab Test Foundation. We've just had a very enthusiastic PhD student getting into this. Wayne Boardman has been a long-term collaborator on this, and of course Peggy Rees-Miller. The zoo has been very supportive, and I've had great discussions with Chris and Mike about taking this further, and Belinda Turner have been very grateful and collaborative at Perth Zoo, and that shows that I'm not only being in the lab, but I also like going out in the field and handling an echidna. And I'll stop there and thank the organisers for giving me an opportunity to present this, and I'm happy to take questions.
We are